my mom.
Flashback.
What's wrong, Mom?
I'm hungry. Food.
Alright, I'll get you some food. Slow.
A few moments later.
You look ugly.
Here, Mom.
Solely. Mom, what is this? She's the nice girl. She's the nice girl who always visit me.
What do you mean, Mom? She's your granddaughter.
I'm your granddaughter. What? No, you're not. No, she's not.
We always visit you together.
Yes.
Oh my god. We need to search doctor right now.
End of flashback.
Now I'm going to say about temporal lobe. So temporal lobe is part of the brain that are on the side of our head. So basically, temporal lobe helps us with three main things, which is recognizing what you see, handling emotions, and also how you behave around people. Okay, first, recognizing what you see. So this temporal lobe doesn't actually help you see. Your eyes will do that, but it helps you recognizing it. Let me give an example. When you see your mother, your eyes will help you see the faces, but this part of the region will tell you that, oh, this is your mother, oh, this is your father, oh, this is your boyfriend, girlfriend, blah blah blah. So when you see someone, you're not just seeing a face, you're actually recognizing it, who the person is. Next is emotions. So inside the temporal lobe, there is a small part called amygdala. This amygdala helps you feel emotions like anger, fear, and also happiness. It also connects feelings to memories. That's why when you look at certain place or people, you feel in a certain way. The last function is social behavior. So the temporal lobe helps you to understand signals from other people, for example, like their facial expressions and the way they talk. This helps you to understand how someone is feeling and how you should respond to them. So basically, it helps you to get along with others. Now, you guys might be wondering, what is the neuropathology associated with focal anterior temporal lobe atrophy? So I want you guys to imagine the anterior temporal lobe is like a busy neighborhood. So this neighborhood is responsible for recognizing people, understanding meaning, and also connecting emotions to faces. So one day, something goes wrong. The neurons. Inside each neuron, there is a protein called TDP-43. This protein usually harmless, but in this case, it's misfold. So at first, the brain tries to clean up the mess, the misfolded protein, but because the misfolded protein keeps increasing, the cleaning system cannot keep up. As more and more TDP-43 builds up, the neurons begins to break down and die. So here's the main problem. Once neurons die, they cannot regenerate back, they cannot grow back. So with many neuron loss, the neighborhood, the anterior temporal lobe, begins to shrink. This shrinkage is why we call it focal anterior temporal lobe atrophy. And because this part of the brain helps us recognize familiar faces and understand who people are, this person will begins to lose these abilities. So they may look at their own family and feel nothing familiar, like they cannot recognize them at all.
Frontotemporal dementia is an umbrella term to describe neurodegenerative disease that is affected in the frontal and temporal lobe. There are three main clinical subtypes. The first one is behavioral variant FTD, BVFTD. It affects personality, emotions, and also social conduct. The second one is semantic dementia, which we also call as SVPPA. It affects semantic memory and language. The third one is progressive non-fluent aphasia, which we also call as NFV-PPA. It affects the speech production. So in this video, we will be focusing on BVFTD and also SVPPA. The main brain regions for this symptom is medial prefrontal cortex, anterior cingulate cortex, orbitofrontal cortex, and also prefrontal amygdala connection. Now we're going to see the neurotransmitter systems. The first one is serotonin. The neuron loss in Ref nuclei and decreased 5H1A and 5H2A receptor binding cause the social misconduct. The second one is GABA. Reduced GABAergic interneurons, particularly calbidin positive neuron, cause the cortical overexcitations, and it decreased intracortical inhibitions. The third one is dopamine, degenerations of dopaminergic neurons and lost dopamine receptor. The last one is glutamate, the reductions of glutamate levels and loss of AMPA or NMDA receptor. Next, we're going to see the cellular pathology. FTLD tau and FTLD TDP aggregates to form intracellular inclusions. This causes the neuron loss, astrogliosis, disrupted inhibitory control, and causing the astrocyte activations and gliosis. The damage in the frontal limbic circuits explains impulsivity and inappropriate social behavior, while the damage in the salience network explains the failure to assign salience, contributing to these inhibitions. The dysfunctions for the executive control networks leads to poor executive regulation.
Now I'm going to explain about prosopagnosia. So prosopagnosia happens when the brain area for recognizing faces are damaged. The first one, we have the right fusiform gyrus. This helps us see and recognize faces. If it degenerates, the person can't recognize familiar face. Next one, we have the right anterior temporal lobe, which stores information about who the person is. If it's damaged, they can see a face, but they cannot recognize whose face it is. Then the third one, we have the hippocampus. The hippocampus area links faces with memories. So when it's affected, the face doesn't feel familiar. And the last one, we have the orbitofrontal cortex, which give emotional meanings to faces. Damage here will make social recognition even harder. And now, the pathway. The first pathway is ventral visual stream. It connects from the occipital cortex into the fusiform gyrus, into the anterior temporal lobe, and lastly into the orbitofrontal cortex. This is responsible for objects and face identity recognition. So if it's degenerate, it will lead to prosopagnosia. And the second pathway is the inferior frontal occipital facilis, IFOF, and uncinate facilis, UF. White matter strikes linking visual temporal regions to frontal emotional centers. If it's damaged, there will be loss of integration between visual recognition and emotional or social meaning.
For the cellular pathology, there is TDP43 inclusions, neuron loss in cortical layers with gliosis, glymphatic system dysfunction causing the impaired clearance of tau or TDP43 aggregates and causing the progressive cortical thinning in temporal regions.
In prosopagnosia, some neurotransmitters don't work well. Glutamate is reduced in ventral temporal cortex. That makes it harder for the brain to process and encode faces. And for acetylcholine is also reduced. This causes small problems with attention and memory during face recognition. So the brain not only loses the face pathway, it also loses the chemical support needed to recognize faces.
The main brain regions include orbitofrontal cortex, medial prefrontal cortex, and right temporal lobe. The salience network dedifferentiation caused the failure to detect social cues, emotional blunting, and inappropriate behavior response, while the executive control network impairment caused the cannot stop, suppress automatic or impulsive actions, fail to learn from social feedback. The loss of serotonergic neurons and reduced 5-HT1A and 5-HT2A receptor binding. The second one is the reductions of dopamine cause the apathy and loss of motivations. The third one, the reductions of GABA cause the excitation inhibition imbalance in the cortex, and the acetotoxic neuronal damage and behavior disinhibitions. Now, the cellular pathology. The BVFTD shows neuronal degenerations and astrocytosis and intracellular inclusion, causing the disruption of neuronal homeostasis and leads to synaptic loss, microvacuolation, and cortical thinning. The white matter hyperintensities reflect axonal degeneration, demyelinations, and these lesions impair communication between the OSC, temporal lobe, and limbic structure. There is many key difference between the BVFTD and SVPPA, but you can read it in more details on this table.
In FTD,
The damage starts in frontal and temporal lobes, which are responsible for emotional, behavior, and social understanding. Meanwhile, in Alzheimer, it starts in hippocampus, which mainly focuses and controls memory. What are the early symptoms of FTD and AD? Alright, in FTD, we can see changes in personality, disinhibition, loss of empathy, and also social inappropriateness. Meanwhile, in AD, it's just memory loss and confusion. In FTD, prosopagnosia or problem in face recognition is very common. Meanwhile, in Alzheimer's disease, it is very rare and it often appears in later stage. In Eleanor's case, she shows personality change and couldn't recognize faces. So, both are early signs of FTD and not Alzheimer. Now, memory and cognition. Unlike Alzheimer, people with FTD remember facts or events, but they act inappropriately due to loss of social controls. Memory in FTD often preserved in early stages. Meanwhile, in AD, the memory are always impaired early. FTD usually starts from the age 45 years old to 65 years old. Meanwhile, in AD, it usually starts after 65 years old. Both FTD and AD involve abnormal protein buildup, but the brain, the type, and also the brain areas that are affected are different. Our neurons make different types of protein to keep the cells healthy and working properly. But in dementia, some proteins become abnormal because they either clump together or they build up where they shouldn't. So these two events causes brains to damage and making them die. In FTD, there are usually two main types of protein that are usually found. The first one is tau protein and the second one is TDP-43 protein. Normally, tau helps to stabilize the internal structure of the nerve cell. However, in FTD and Alzheimer's disease, tau becomes abnormal and sticky and forming tangles inside the neuron. When this happens, neurons cannot transport nutrients properly, it weakens and eventually dies. TDP-43 protein normally helps to control how genetic information is used in cells. However, in some FTD cases, it clumps up inside the cytoplasm, where it shouldn't be. Therefore, it causes more damage to the cell. Therefore, depending on which protein is the main abnormal protein, FTD can be called FTLD-tau or FTLD-TDP. In Eleanor's case, the first symptom was not memory loss. Instead, she showed major changes in behavior. She became disinhibited, she lost her social manners, and also couldn't recognize familiar faces. This is different from Alzheimer's disease because Alzheimer's disease patients usually start by memory loss. For example, forgetting names or daily events, while their personality remains mostly normal at the beginning. So, the main difference is where the disease actually starts in the brain. In Eleanor's case, the frontal and temporal lobes would affect Eleanor's behavior and recognition first. Meanwhile, in Alzheimer's disease, the hippocampus lobe would affect memory first.
In this slide shows the difference between BVFTD and normal aging. At first, primary location of BVFTD is at the frontal and intertemporal lobes of the brain. This part usually control behavior and personality. This BVFTD also have often asymmetric pattern of the brain, so which means one side is worse than the other. Also means that it is focal and disproportionate. So in early stage of BVFTD, usually hippocampus is relatively spared, helping us to differentiate between Alzheimer's disease. On the other hand, normal aging is global, symmetric, and generalized. Let's move on to the treatment for FTD. Currently, this disease is no cure, so we manage to focus on therapy and medication. For medication, we usually use SSRIs like citalopram to help manage the mood symptom and common behavior. Next, we avoid cholinesterase inhibitor such as donepezil, which are typically used for Alzheimer's disease. If you use to the FTD patient, it can worsen the FTD patient. Next, on the therapy side, we have speech and language therapy and occupational therapy. For the speech and language therapy, is the key for the language variant and focus on compensatory strategies. So for occupational therapy, it's also essential to maintain the function and independence for as long as possible. Let's move on to the long-term outlook of FTD prognosis. So the expected life of FTD varies by subtype. So an average survival for this disease typically 7 to 13 years. So next, the disease cause involve progressive neurodegeneration. For example, in the semantic dementia, at the selective atrophy at the anterior temporal lobes will worsen over time. This lead to the complete loss for the semantic knowledge, result in agnosia and anomia, which means inability to recognize object and name them. Last, in the later stage. So the disease often spread and leading to severe behavior and intellectual disability. So include mutism and have motor difficulties.